So I'm a new researcher at the Agricultural Research Organization, Volcani Center here in Israel. And I have a confession to make. I'm not a plant scientist. I have literally no background in plants. I did my postdoc in material sciences. But I'm here today to present a method that I think can be cool in investigating some plant microorganisms interaction, and to hear your feedback and to hope that you will agree with me. My background is in biomimetics, and the method I'm going to use is from Biomimetics. Why is this happening? So the first thing I'm going to do is explain to you what biomimetics is, and then I'm going to show you a little bit of data from several projects that we have, just to tease you a little bit. So I'll start with the leaf, because the leaf is a more stable system in our lab. I'm then going to show you the challenges that we do have in this method with the root, and then I am going to show you some results that we have with the root system. So what is biomimetics? In order to explain that, I'm going to start with an example. So if you ask any person in the biomimetic field, like, what is the example for biomimetics, this is the example they're going to give. This is the lotus flower, beautiful flower. But it's actually not the point. The point is the lotus leaf. So the lotus leaf has this property of being super hydrophobic. And when researchers were interested in that and investigated why is it super hydrophobic, what they saw is that it has very little to do with the chemistry on the surface and actually a lot to do with the structure of the surface. So this is the microstructure of the lotus leaf, which is what causes its super hydrophobicity. That intrigued researchers and brings me to what biomimetics is. So biomimetics is using chemistry and material sciences to mimic biological systems. The general logic being nature has an advantage of billions of years of evolution over us. Nature had solved a lot of problems with this process of evolution. So why not look at nature and get some inspiration as to some of the structural solutions that nature got and try to mimic them for our own advantage. So this is being used a lot in architecture and in design, which is less my field, but also in the field of microstructures. So as I said, the first example that comes to mind is the lotus leaf. So once people discovered that the superhydrophobicity is caused by the microstructure, they said, okay, let's mimic this microstructure to make superhydrophobic surfaces, and they will be self-cleaning surfaces. Another cool example for this is the gecko. This is this annoying creature that walks on walls. So it's able to walk on walls because it has glues. It has the glue, but the real reason it sticks to the wall is actually the microstructure in the paws. Another cool example is the spider web. So those are very tiny but very strong fibers, and this again has to do with their microstructure. And then butterfly wings, or feathers really in general, many times they're not, the colors that you see in nature many times are not generated by pigments, but rather by micro and nanostructural features that absorb specific wavelength and don't absorb others. So biomimetics is generally taking inspiration from the structures and mimicking them to benefit mankind. But in my lab, we thought about taking it to something a little bit different. Why not take those structures, mimic them, in order to learn something about nature, actually take it back to nature? And what do I mean by that? If you look at plant microorganism interaction, actually any interaction that the plant has, the surface of the plant is the entry gate. Okay? This is the first thing that the microorganism would see. A lot of interactions occur on the surface, and you all know that. So we have interactions on the leaf and interactions in the root. Some of those interactions, as we've heard today extensively, can be beneficial to the plant. Some of those interactions can be harmful. However, while those interactions have been studied, in these interactions there are a lot of signals that take place between the plant and the microorganism. And there are many chemical and molecular signals that are widely studied, and one signal that is a little bit more ignored, and that's the physical signal. The topography itself, the microstructure itself, is actually a signal to the microorganism to know where it's at and if that's a place it wants to be in. But the problem is that when you study those interactions on the natural system, it's very hard to separate the chemical molecular signals from the physical signals, because biology is hard and it's very complex and it has a lot of noise in it. So we were wondering, could we use biomimetics to actually separate those two signals? So what we do is we make those synthetic surface microstructures. We use a method that is called soft lithography, but it's really just molding. So for example, with the leaf, you would take the leaf, you glue it to a Petri dish, you will then pour a polymer. The polymer is liquid, it will capture the structure. You then cure the polymer, it solidifies, and when you remove the leaf, you get the mirror image of the structure, or if you will, the negative. So in order to get the actual structure, all you have to do is repeat the same process with a negative. But the cool thing about it is that the negative does not get destroyed during the process, so you can actually repeat the experiment with the same exact structure over and over and over again. So this is what it actually looks like. This would be an actual leaf. We work with tomatoes, and this would be a replica of a leaf. And this is what it looks like from the inside. So this is the leaf, this is the negative, and then this is our positive replica, so we can recapitulate the vasculature and the hairs, and also the cells and the stomata. So this technique has a pretty high resolution. Okay, so we thought, what can we do with this method? And we're working in collaboration in several projects with plant pathogens and see how the structure affects the interaction between the plant and the pathogen. So the first pathogen I'm going to show you is Botrytis. So Botrytis is this horrible fungus that does horrible things to tomatoes, and we don't like it. If you look at an SEM image, you can see the spores of the Botrytis, and you can see it's roughly in the same order of magnitude as the microstructure. So it actually makes sense that the microstructure will affect what the spores are doing. So what Helen in my lab did, just to show how that works, instead of molding the positive on our regular polymer, she just molded it on an agar plate, the same agar you use to grow the fungus. So she took two non-structured plates and two plates with structures, and she just spread a little bit of fungus in the middle. And what she saw in day 3 is that while the colony of the fungus spread like a circle in the non-structured plates, it spread it across the veins in the structured plates, and it, of course, continued into day 5. And it's interesting because there have been reports of fungus appreciating the vasculature area in the leaf. And people have said, well, of course it likes it there because this is where all the food is. But here, those two surfaces are completely identical food-wise. It's just agar. It has food everywhere. But still, it gets a signal from the structure itself. It knows how to walk along the structure. Another example I want to give to you is from Calla Lilies. This is done in collaboration with Didia from my institute. So Calla Lilies are beautiful flowers. They're colorful. They have white and other colors. But they also suffer from diseases such as soft rot caused by this bacteria. And what Iris saw was that the white lily is actually more resistant to that bacteria than the colored lily. And when she looked at the structure of the leaf, she saw that the white lily leaf is smoother than the colored lily leaf. So this is how the leaf of the lilies look like. But these are actually our replicas and not the natural leaves, but they look exactly the same. So then she took the replicas and she put some bacteria on it, and if you have a closer look, what you can see is that while on the white lily leaf replica, the bacteria are on their own, On the colored one, the more rigid structure, you can see how they are clustered together and starting to form a biofilm, which is exactly what she sees in the natural system. But again, this is not the natural system. This is a synthetic surface, and both of the surfaces are completely identical chemically. The only difference is the structure. So we are interested in moving this method into the root system. The problem is that the roots are significantly harder to work with. First, they're not flat sheets like leaves, so they're actually three-dimensional. And second, they're much more delicate than leaves. So our first attempt in replicating the structure of the epidermis, we're looking only at the outside part, was just doing exactly what we did with the leaf. That was our first attempt. So we used the exact same polymers. This is the Polymer Pdms. It's a silicon oxygen polymer. It's very convenient to work with. It's biocompatible. It's transparent, it's commercially available, and fairly cheap. And these are the best results that Neta from my lab got, which are not bad, but are not the best, I think. You can definitely see how we can generate the cells. We can see a lateral root emerging, and we can capture the root hairs to some extent. Root hairs are actually our biggest problem. And are kind of an important structural feature to just give up on. So that was our first attempt. And then we thought, okay, if that doesn't work, maybe we can use a different polymer for the negative. Maybe the root hairs will not stick as much in something that is a little bit softer. So we tried to use gelatin, which is a water-soluble protein, and it's significantly more soft than PDMS. And these are the results that Paravi got with the gelatin, and as you can see, not as good. So we said, okay, using a soft polymer for the negative doesn't work. How about using something that is significantly harder? And this is what we did here. We used polyurethane acrylate. And the cool thing about that is that it's completely liquid. You just submerge the root in it, and then you shine UV on it, and it polymerizes, and it becomes really, really hard with the root emerged in it. So this is not perfect yet, but this is the negative replicas that Paravi got, which you can see are much clearer and better, and we can actually also get the hairs in the negative. The positive still isn't working that well. Again, we can see a very good cell structure, but we still can't get the hairs, but we're getting there. In the meantime, we're trying the system that we already have with two systems. One of the systems is Ralstonia, which is a soil-borne bacteria, which is horrible for tomatoes. We're working in collaboration with Angeline Purdue, and what she saw is that if you have a resistant and a susceptible plant, the bacteria colonizes those two plants completely differently on the surface and in the vasculature. So the first thing we're doing now, and this is very much work in progress, so Tali is looking at the structures of the resistant and the susceptible plants just to see if we can actually see by eye if there is any structural differences. We're not there yet, but again, working on seeing structural differences. And then one thing we did do is we sent to Angeli two replicas, one replica of a needle, so basically a smooth surface, and another one a replica of a non-perfect root, but nonetheless of a root. And you can definitely see how the bacteria colonizes the two surfaces completely differently. And the last one is root knot nematodes, nematodes that we just heard about, different nematodes, but still. So they are also horrible for many crops, specifically tomatoes. And our collaborator found that they do like to penetrate in a specific area of the root, in the elongation zone. And we basically asked ourselves, is there a structural component for this preference? And so what Neta did is she took, again, a replica of a needle, a replica of a root and a natural root. She just dumped a few nematodes on it, and after a while looked at it, and she saw some nematodes stayed on the root, some stayed on the replica, but she saw none on the needle replica. And basically what we think is that when the nematode is walking on this root or replica, when it sees a structure, it's like, oh, okay, maybe there's something in here, but on the needle, it immediately it sees that it's been fooled, and it just goes somewhere else to look for food. So with that, I'd like to summarize. Surface microstructure acts, or I want to convince you that it acts, as an additional signal in plant-environment interaction. There isn't just chemical and molecular signals, but the structure itself plays a significant role in signaling. Our goal is to separate that physical effect from the chemical, a molecular effect. The tested pathogens that I showed you show structural preferences in both leaf and root. And we're using biomimetics in a completely different way. Instead of using it for mankind, we're using it to go back in nature and ask questions about how nature works. With that, I'd like to acknowledge all the people who did the work. I showed them during the presentation, all of my collaborators, funding. This is my website. We're always looking for eager and determined students, so feel free to approach me, and thank you for your attention.